Yeah, so Robert went there, and I heard him tell a story yesterday at lunch, and he said that he arrived there as a very much cardiac person, and they just gave him freedom to follow that functionology and connectionology and develop his own space there, and with a lot of support. He made that transition from being a mathematical modeler into being a molecular biologist, and he stayed in that space ever since. But he made some very important contributions while he was around that, in particular identifying the role of the microtubule network in Actin connections to the membrane, the cardiac cells, and their role in the generation of Actin isofunctional distributions out there. Since then, he got a job at University of California, San Francisco, faculty at the Cardiovascular Medicine. Did very well there. Attracted some very talented investigators, amongst them Jamie Smyth and Sammy LaRue, who Sammy also worked with Robin in a biotech company that ran up. And of course, you know how successful that story continues to be. Since that time, Robin was recruited down to Los Angeles to work at the Cedars Sinai. He was recruited by Eduardo Marbayan, who's one of the most prominent figures in the field of cardiovascular biology. He was the former editor of Biomedical research, and a very well-liked and respected scientist, and also academic in the sense that circulation research is very well understood. So Robin's one of the number of PIs who are now at Cedars Sinai and continues to do fantastic work. Given that he's an MD PhD, he's a very competent individual that we hope to complete for our new building.
That was a fit.
He's basically vocationally aligned. Our one time, about transplant, so really one who's getting on the way of working with the postdoc and having these things. I think Rob has a massive success with that. So I'll leave it over to you.
I think we're good. Yeah, I got to say that's the most fulsome introduction I've ever received. I'm coming back. But really, it's a story of being lucky enough to be in the right place at the right time, and learning from really sort of those who have done it around me. And if it's a story of anything, it's a story of being able to learn from the right people. And it's incredible to be here, because not only is this the most connected, intense place I can ever imagine. I still think it beats the New York folks. So literally anywhere. But what's developed here is really the dream of not just physicians and medical centers, but of scientists and of where the NIH wants things to go. And what's just remarkable here is not just the biology, but the sort of cell-to-bedside translation, or that you're in the process of, is really not to be underestimated. And I know you're very understated about it, but I really think that the future of connection biology and with heavy implications to cardiac and other systems is really going to come from here. You can imagine, for that reason, I'm very intimidated to be here. So with that, I'm going to put out a couple of stories that we've been working on, try to give some perspective early on. And really, we're just trying to pull together the pieces. This is very much in development. Eager for your feedback. And so the title of the talk is GJA1-20k, Functions of the Cardiomyocyte Stress Response Protein, and I hope I can convince you at least of that, with all that implies. But just by way of background, and I'll move through a little quickly with a group like this. Gap junctions really started making their name in the heart, where they're essential to the electrical activity of the heart, right? So there's 3 to 4 billion cardiomyocytes in the ventricle. They need to contract near simultaneously. The way you get very rapid spread of excitation is cell-to-cell coupling via gap junctions. And in the bottom is, of course, isolated mouse ventricular cardiomyocytes, primary antibody against connectin 43, and the connectin 43 localizes to the intercalated disc regions at the end to end. And we know why that occurs, right? We need rapid excitation down the longitudinal direction of fibers. But how it occurs is a different story, and that's not just a academic question. This is on the right is a slide it's already over 20 years old, from Andy Witt's group. And he did what ended up being a classic experiment. He took dogs, he tied off their main coronary artery, the left anterior descending artery, and he essentially created a heart attack. He sacrificed the animals 4 days later, and then he just did connectin 43 staining, and you can see in the regions directly fed by the LAD, there's no connectin staining, essentially there's scar. In the regions well away from the area that's fed, you can see a distribution, localization of connectin 43 at the cell-cell borders. Importantly, in between, in this so-called border zone, the connectin 43 is actually off the intercalated disc. It sometimes appears laterally. It also collects inside. And so this is a change in localization, a decrease in coupling at the cell-cell borders that has significant implications. This is just, I thought for fun. You know, this is already, 25 years ago? Even more than 25 years ago. And some of the modeling work we're doing with Yoram, this is simulated tissue, okay? 100 by 100, 10,000 cells. Perfect, literally, complement of ion channels and gap junction coupling that's completely symmetric, all right? Because it's a computer model. And what you see is the color coding here is blue is resting membrane potential. As you get hotter, you get towards peak potential. And if you introduce a line stimulus, see if I can do it here, whoops. Introduce a line stimulus, you see a quick depolarization. If we introduce a premature stimulus from the side, what you see is the start of what seems to be an arrhythmia and it self-terminates. So in well-coupled tissue, an extra beat is simply that. It's a premature stimulus that terminates. It's seen as on an EKG as an extra beat. It's completely benign. It means nothing clinically. But if you just reduce the coupling by 50%, and that's not atypical of ischemia, and you repeat the protocol. When we repeat the protocol, what you get is a reentrant arrhythmia, okay? And that's the equivalent, the EKG equivalent of going from a sinus rhythm to what's called a monomorphic ventricular tachycardia. Monomorphic ventricular tachycardia is dangerous because it often degenerates into ventricular fibrillation, all right, or sudden cardiac death. You can see even in perfectly symmetric tissue, a model, you can see degeneration into a more disorganized pattern, okay? So here you have, all you have to do is reduce the coupling you leave the other ion channels intact, and you get profound changes in the electrophysiology. And it is the coupling that is most significantly changed during episodes of ischemia as well as in heart failure. So the issue comes really down to how does this coupling reduce? What maintains it normal? What causes it to be reduced? Studies by folks in the room, as well as others, have really identified that the gap junction half-life is hours, right? 4 to 6 hours. And so when we talk about turnover, when we talk about the need for proteins to be where they need to go and how that changes over minutes to hours, we're talking about trafficking, okay? And to convey the point about trafficking, this is from my days in San Francisco. Of course, you recognize the Golden Gate Bridge, right? This is the Marin side. This is the San Francisco side. And normally, it takes, the bridge does its job. It's there 365 days a year and 24 hours a day, and it takes just a few minutes to cross the bridge by car, maybe 15, 20 minutes by bike, a little bit longer if you walk it or jog it. For me, that's about 3 hours. And but the point is that even though the structure of the bridge is there, no matter what, it only takes one perturbation, and that perturbation can be a rock concert or construction or an off-ramp closure. And with that perturbation, you get gridlock. So when you have a perturbation in the traffic, and especially the apparatus that performs the trafficking, the rest of the apparatus can't do what it needs to do, which is get the protein of where it needs to go. And so just in about 30 seconds, a history of trafficking in general, really started with George Pilati, right? Who was in the 50s through the 70s using EM to identify the structures of the cell and the organelles. And he really identified most of the major structures from nucleus to ER to Golgi, and even started identifying these intracellular vesicles that bring proteins to where they need to go. And then really it was Gunther Blobel and his colleagues at the Rockefeller who brought to us the concept of zip codes, right? Protein recognition motifs. So when the protein was where it needed to be, when it was at its destination, it knew it was home. Although, frankly, the question that hasn't been addressed is how does the protein know how to get there? Just recently, Jim Rothman and colleagues were recognized for really showing us how the intracellular vesicle moves, as well as how it fuses. But again, how does it know to get to its destination is a question that still exists, and it's a very important question given the fast turnover of these proteins. So we've been focused on this question for a better part of a decade. And I'm trying not to summarize too much of this work in 50 minutes. But essentially what we've developed is what we call the targeted delivery paradigm. And that is when a channel comes out of the ER Golgi, it knows where it needs to go. And in fact, it can go, specifically a connexin molecule can go directly to the endonucleated disc. A calcium channel can go directly to T-tubules. And the question we keep asking and are chipping away at is how does it get there? When we initially defined, this was back when I was in the Jan lab, what we now call targeted delivery, we identified 3 components. One is the specificity with the cytoskeleton delivery apparatus, with the microtubules and plasma tracking proteins at the end of microtubules, EB1 included, okay? Number 2, a big part of the specificity was the membrane anchor, all right? N-cadherin complex, been one for calcium channel delivery to T-tubules, and that's where we spend most of our time. And then number 3, we mentioned, but frankly had no idea how it worked, is there's something about the channel itself. There's something about the membrane protein itself determining its own location. And it really wasn't until a discovery with Jamie, by Jamie, that really helped us understand how that channel can apply specificity. So if I can do anything today, I can convince you of how the channel can help determine its own specificity of where it needs to go. And so I actually took out some of the slides describing this because I assume you know most of the story, right? But really, you know, Jonathan Weissman, who was at UCSF, and Jamie, I think it was honestly, there's a seminar or a lecture you heard from one of his postdocs, right? So Weissman really has made popular the technique of ribosomal profiling. Basically, you know, you fix ribosomes in the process of translating, and in an unbiased way, you basically sequence the mRNA that the ribosome is attached to, and then you can just do statistics on what sequences ribosomes happen to hang out on. And the understanding that the sequence that they most likely dwell the most time on is when they initiate translation. That's the dwell point. And so what Weissman has identified, which the prokaryotic biologists have known for a long time, is yes, on the initial start sequence, on the initial mRNA sequence, ribosomes initiate translation. But there are downstream sites that ribosomes tend to dwell on as well. And those downstream sites map out to methionines, map out to AUGs, okay? And so in brief, ribosomes can initiate translation of a single mRNA molecule, not just at the start of the mRNA, but downstream, at downstream methionines. So then Jamie really said, hey, I wonder if that happens to connection, okay? So we, like everybody in the connection field, would always do a Western blot, and we would chop off whatever was downstream of our 43 kilodalton band. And so Jamie looked at a full band, this is of human heart, and really just mapped out, if you looked at the smaller bands, and they did a simple, you know, sort of in silico analysis, the predicted sizes from the methionines, it was a pretty fair match that the smaller bands could potentially match out to internal translation start sites. And you know the rest of the story. It turns out that's the case. In that, you know, one of my favorite Western blots said, I literally almost fell off my chair when Jamie brought this in. He did it in about a week. Am I putting spin on it, or is that a fair? Yeah. And I still get goosebumps. Literally, when I show this blot, and that basically started knocking out internal methionines. And what you saw is the so-called non-specific bands below the 43 kilodalton band. They disappear and come back when he puts them back one after one. Okay? So these internal methionines are internal start sites. Okay? And I've taken out the rest of the proof where Jamie really established that it's not a product of cleavage. It is truly in-frame internal start sites that occur from a single connection 43 mRNA molecule. And of course, connection 43 has a single coding mRNA, so it's not a splice. There's a single coding exon. So the question then comes, okay, if there are smaller guys, which we've subsequently come to name based on their size. So the one that's most prominent that we focused on, for reasons I'm going to show you, is 20 kilodaltons. We call it GJA1-20k, where GJA1 is the name of the connectin 43 gene. And so the question came in, what do these guys do? All right? So if they can be produced endogenously, what function do they have. And so here's a really a simple experiment where Jamie's plated cells, right, and just quantifies plaque between cells. How many cells had a nice clear plaque between two cells, connections in green. And when he started knocking out the internal methionine, he knocked out the first internal methionine, and there wasn't much change in plaque. He knocked out the second as well, not much change. The third, you get a statistical drop in the amount of plaque, but frankly, you know, not much to shout about. He took out the fourth methionine, the so-called 21 that corresponds to a 20 kilodalton fragment, which is essentially the length of the C-terminus. That starts out at the 20K. It's just as you're coming out of the last transmembrane domain. So the whole floppy C-terminus, and the plaque goes away. And the rescue experiment works, okay? In that knocking down all the endogenous connexin 43, giving back what we call then connexin 43 and 4L, essentially giving back the full-length protein incompetent of the first form of thionine, and then with a plasmid that just expresses the 20K can restore connexin 43 at the cell-cell border, okay? So, you know, think about that. It's not the full-length protein that's needed to get the full-length protein at the border. It's the smaller guy. It's the 20K isoform that's necessary to get connexin 43 to the cell-cell border, okay? And so I'm not going to show you the data for reason of time, but Jamie went on to show that this process is mTOR dependent, or in fact, when you inhibit mTOR, you get increased expression of the smaller isoforms. And the point there is that internal translation is not just an evolutionary phenomenon. It's most of the time, it's been studied, you know, when it's studied in eukaryotic cells, it's been studied in yeast. And how does the genome change its expression of an existing gene, you know, over generations? But once you start bringing modification of mTOR, and subsequently other groups have introduced other pathways that affect it, including MINK-K1-2, the bottom line is when you start relating the change in expression levels to pathways, that implication is metabolic condition can change the expression levels. So this is where a gene with a single coding exon can actually change its product, not through splicing, but through the way the ribosomes translate the mRNA. And they're smaller subproducts of the gene that truncated isoforms, if you will, that can either facilitate the full-length protein or may have other roles in the cell. Okay? And so when we talk about it in a larger perspective, classically, just as I introduced, connexin 43 is a cell-cell coupling protein important to the heart and other organ systems. But there's a plethora of other roles of connexin 43, from trafficking itself to other ion channel trafficking. These are associations that have been reported. Wound healing, out of respect to my local host, ischemic protection, muscle differentiation, gene regulation, cell cycle regulation, cancer progression. Really pretty much most things that cells do, connexin 43 has been associated. And yet it's hard to explain a lot of these functions, how a cell membrane channel can regulate these functions. And when we went down to Cedars, a big part of our agenda was actually to find out what the smaller guys do. And I'm going to tell you a couple of stories. But fundamentally, we found that the smaller isoforms can explain a lot of the phenomena that we see here on the board. Okay? All right. And so one of the things that we have moved to is we've started to incorporate more in vivo work. And so doing the exact experiment that I discussed a few slides ago in terms of coupling, we said, does it actually work in the mouse? And one of the ways to do that is just to introduce the 20K plasmid into a mouse. We do that with package 20K into AAV virus. We do retroorbital injection. Let it. AAV9 has a tropism for the heart. And so 4 weeks later, we're ready to do something to the heart, okay? So 4 weeks earlier, we basically injected virus into the mouse's eye. You know, phenotypically, we don't see much of a difference at all in terms of growth phenotype, all right? And then we do simple histology on a wild-type mouse, right? And this is using connexin 43. We're looking at N-terminal antibodies, so we're looking really at, you know, in endogenous protein. And what we see, we have two negative controls. And one negative control is just a simple GSD plasmid, and the other is 43K. This is 43. This is the full-length connexin 43 molecule, the channel protein, if you will, that's incompetent of making the smaller isoforms along the way. All right? And what you see is that when we introduce the 43K, we do see more signal. You know, from your perspective, just, you know, squint a bit and you'll see there's just more signal here than up here. But in general, we don't see it at the plaque. You see it distributed really throughout the cell. Whereas when we add the 20K plasmid, to the point where we can barely see it on a Western blot. We see it clearly on imaging, but we can barely even see it on a Western blot. Yet, look at the richer plaque that's formed, and the red is a cadherent, so this is the merge signal. The bottom line is, when we add the 20K plasmid, we get more plaque at the cell-cell border. We get more connexin 43, endogenous connexin 43 plaque at the cell-cell border than when we actually add the full-length protein. And that's borne out biochemically as well, using sort of the triton solubility assay, right? We can differentiate between so-called soluble or how we like to think cytoplasmic connexin and insoluble, which is more in the plasma membrane. And what we see is just what we saw in the imaging, is that you know, when we add connexin 43 in the mouse 4 weeks ago, we then see more connexin 43 total really in the cytoplasm, but actually when we talk about the plaque, we get more connexin 43 at the plaque. This is endogenous connexin 43 at the plaque than if we actually add the full-length protein itself. Okay? If you add the smaller guy, you get more of the endogenous full-length protein at the border. So this offers a wonderful opportunity to address the trafficking question that we first started. And in fact, we started to change our thinking. It was a decade ago, we were saying trafficking is how does the membrane protein and how does the vesicle carrying the membrane protein choose the right pathway? How does it choose essentially the right train track to its destination, right? You know, what's its postal address? Instead, we're now thinking that actually the smaller isoform, specifically the 20K isoform, can utilize the cytoskeleton to actually pattern the cytoskeleton. And in fact, it's not so much choosing the right track, it's laying its own track, its own delivery track. Here are some of the data. So in another paper by Jamie, this was 2012 in Circ Research, he looked at individual connexin 43 particles within a cell, okay? And microtubule transport is fast, kinesin-based transport. And what we saw most of the time is that the individual packets of connexin weren't moving that fast, okay? Most of the time, they're sort of doing a staccato movement. And in fact, when you quantify that, over 85% of the connexin 43 within a cell is pretty stationary. When it is stationary, it's not necessarily on microtubules. In fact, it's associated with actin. So it's hanging out on these actin rest stops. And so when we added 20K, we were just in the phenotyping mode. We added 20K to cells, and we actually, by fluorescence, we saw a lot of actin. We saw these actin ropes, essentially. Any kind of phalloidin staining, any kind of actin staining. This is not hard to see. And so we looked by EM, and what we saw is I hope you can see it in individual fibers. When we added 20K, we don't see any increase in the total amount of actin. So there's the same amount of actin protein. However, when we look at fibers, and here we've just actually, to make it easier, we've outlined the individual actin fibers, what we see is significant increases in both the length and the number of actin fibers. So we actually have a whole program, frankly, on 20K nucleating and promoting actin polymerization. And so when we talk about actin really being promoted by 20K, and 20K really facilitated microtubule-based connexin 43 delivery. It's just a matter of organizing which comes first, all right? And we believe that the order is actually 20K is organizing actin, which in turn organizes the microtubule delivery highway, which in turn delivers the larger channel. Here are some of the data. First of all, by co-immunoprecipitation, right, to see if these proteins cluster together. You know, to immunoprecipitate and pull down 20K, right? But what comes with it is both actin and alpha-tubulin, okay? So both actin and tubulin, the two major proteins that form the cytoskeleton, really cluster with 20K. And when we look at EB1 trafficking, I don't show here the tip protein, we saw an increase of EB1 going the border. And here we see a sort of interesting experiment. We're looking at tubulin in the images, okay? So this is tubulin. Here we've sort of, just for clarity, we've outlined them, the individual lines. So here's a cell-cell border, and we saw microtubule. These are the highways, microtubule highways that deliver protein to the membrane. When we give letrunculin, when we stop actin polymerization, we lose microtubule attachment to the cell-cell border, okay? When we give the 43K isoform, the eventual cargo, we don't see much improvement. When we give 20K, we actually rescue microtubule attachment to the border. Essentially, by giving 20K, we are improving the ability for microtubules to deliver connexin 43 to the border. And the reason we're doing that is actin is paving the way. Actin is organizing the highway for connexin 43 delivery. And so, you know, essentially what we have in order is we have the smaller connexins, specifically 20K, is organizing the filamentous actin, which then is followed up by the EB1 microtubule growth, which in turn delivers connexin 43 at the gap junction. Okay? So the title of the talk was connexin 20 is a cardiomyocyte stress response protein. In other words, it ticks up when the heart is under stress. When we talk about stress, we're talking about ischemic stress and heart failure stress. And this is some of the data which indicate why, is that when we look at 20K, this is actually another paper that just after Jamie did, also independently demonstrated that there's smaller isoforms and subjecting brain to hypoxic stress in the hypoxic chamber in rat brain, okay? They actually summed up the smaller isoforms and found quite a difference and increase in the smaller isoforms just by Western blot when subjecting brains to hypoxia. We found the same thing, that, you know, we subject hearts to 30 minutes of ischemia, basically no ischemia, or ischemia reperfusion. And what we get is a significant increase in 20K. Just so you know, I don't know if it's worthwhile, we see a similar increase in 43K, subjected to ischemia, but when we do ischemia preconditioning, we just see the 20K increase and not the 43K. And so let's go back to that mouse heart experiment, okay? I should mention to you that actin polymerization is affected by ischemia. So can 20K stabilize things during ischemia? We took the same mouse hearts, okay, from the wild type experiment, but this time, instead of just sacrificing the animals and slicing the heart, we perfused the heart with a Langendorf perfusion, ex vivo, right? And we subjected those hearts to 30 minutes of ischemia. And after 30 minutes of ischemia, we simply then just fixed the hearts and looked at the connectin 43. And so now look, in terms of the connectin 43 signal, okay, this is our two controls, GFT and 43K, and then this is 20K. Again, 4 weeks earlier, they got the 20K plasmid, all right, in AAV9 virus. And that actually, if you look at the connectin 43 signal, you see really a substantial maintenance of plaque at the cell-cell border, which goes away after 30 minutes of ischemia. Okay, so if you have ischemia, of course connectin 43 is downregulated, right? Everybody will tell you that. Look at these mice that have a little extra 20K, and I do mean little. They actually are able to withstand 30 minutes of ischemia. And this is just a quantification. So really, you know, if you get a 30 to 40% drop with acute ischemia, when we give the 20K isoform, we maintain, we restore, we prevent the loss of connexin plaque at the cell-cell border. Okay? Yeah, please, Rob.
I'm intimately familiar with the paper, of course.
Yeah.
But remind me, is the ischemia a global ischemia?
Global. It's not very sophisticated at all. We turn off the perfusion of a Langendorf apparatus.
Yeah,
Yeah.
that's fine. Gives you all the material to work with.
Yeah, exactly. And it's uniform. And so from the trafficking perspective, okay, and you can imagine, you know, sometimes I give this talk, am I talking to scientists or am I talking to clinicians? And so from the trafficking perspective, if you think about it, or if you think about me at Cedar Sinai, okay, here's Cedar Sinai, we live about here, you know, it's kind of hard to get around Los Angeles, in case you've heard. But say I wanted to go to the Santa Monica Pier. Okay, that's what shows up in the, anybody's old enough to see Three's Company, that's the opening credits. What I would do now, literally, and I'm sure everybody in the room is the same way, is if you want to get to the Santa Monica Pier, you'll know it when you get there, but how do you get there, right? Well, you take out your smartphone and you map out a course, and based on driving times, you can get from Cedars to Santa Monica in the best direction. How does a protein do that? If you've got a connectin 43 channel, and it needs to get to the intercalated disc, and it's got a short amount of time to get there before it's degraded, how does it know how to get there? It doesn't have a GPS. And essentially what we're suggesting is that its way is paved, all right? It's paved, its highway, its route is designed by the smaller isoform that actually then helps lay down the highway that gets it to where it needs to go. Okay, so just to switch tracks and try to do this in the next 15 minutes, I hope, if I really succeeded today, when I finish this next story, you'll see the commonality, okay? And so this is a more recent story that's still developing in our group. Shan Zhang, when she was with us, probably one of the best things Shan did was completely ignore what I asked her to do. And that is, you know, I'm so focused on the cytoskeleton, if you haven't gotten that point. And that Shan just, I told you, we're doing expression analysis. Put in the protein, seeing where it ends up, right? And Shan kept saying, Robin, it's in these clusters. And I said, literally, Shan, please, it's about trafficking. Forget the clusters. We're overexpressing protein, whatever. And she actually picked up on some other ideas that were floating around the lab. And she said, I'm going to look at different organelles, and I'm going to look at mitochondria. All right? And so to cut things short, this is where this is going, is that Shan co-stained with mitochondria, and she found something which was startling. This is, let's start with a negative control. We'll use 43 kilodalton as a negative control. In an HEK cell, all right, this is a mitochondrial marker. And what you see is a 43 kilodalton protein. Yes, it kind of associates with the mitochondria. You know, does it preferentially localize there? That's sort of a hard argument to make. All right? This is the 20K isoform. All right? And this is just a blow up, but this is the full cell. This is a mitochondrial marker. This is the merged image. Okay? It's one-on-one. Is this a fixed cell artifact using live cells, using the TMRM, viability stain of mitochondria? This is the 20K isoform. It's one-on-one. Okay? And we've tried this in a number of primary cells and in cell lines. And it's uniform. Okay? So in our hands, we haven't found a situation where the 20K isoform, this trafficking isoform that all we cared about is trafficking, was actually had a tropism from the mitochondria to the point where it's a mitochondrial marker. And this is just a biochemistry. We could do a Perkle gradient to separate it. But essentially the story is that while this is on your left is 43 kilodalton protein, on your right is 20K. And you can see that, you know, as Tom20 is enriched, 43 kilodalton protein, especially in the F2 and F3 fragment, really doesn't appear. It's mainly in the fragment that the mitochondrial marker is not in. The opposite is true for 20K. As you get enrichment of Tom20, you get a good amount of mitochondrial 20K. And so, so that's biochemistry confirmation. And when we go back to EM, we did immunogold labeling, okay? And so we sort of labeled the 20K in HeLa cells, just overexpressed HeLa cells. And what we see is that 20K tends to circle microtubules. It also loves to hang out at mitochondria. And we actually quantified that, right? And so, you know, 5% end up in the Golgi, not quite half end up in the ER, okay? Fully 63% of the immunogold dots that we counted were associated with mitochondria. Fully 15% were associated with microtubules. And, you know, you're probably doing the math saying that's greater than 100%. And it is greater than 100% because they occur together, okay? 30% of the immunogold particles were at interfaces that we could tell of mitochondria ER, and another 10% at microtubules and mitochondria, okay? That was a GFP, yeah. So we would use our anti-GFP. That's exactly. And so for the immunogold labeling. And so what this tells us is that, you know, just what we're seeing by fluorescence imaging, it hangs out at mitochondria, and in fact, it hangs out at interfaces with the microtubules. And we did see it also, there's a cancer cell line that expresses quite a bit of the endogenous 20K, and so with that cancer cell line, without labeled protein and without overexpressing protein, we did see it also surrounding the mitochondria, endogenous protein. But the association between mitochondria and microtubules, obviously is an aha moment for a trafficking group, right? Because there is a growing literature on how the mitochondria use the cytoskeleton to distribute around, right? And so one of the ways we address that is that, as this group knows better than anyone, that there's a putative binding domain, microtubule binding domain, on that connecting 43C terminus. It's pretty high up, right? And there are actually 6 key amino acids that mediate the binding. And we've done co-immunoprecipitation experiments that actually we created a mutant 20K that deletes those 6 amino acids, all right? And that mutant in the co-IP assay certainly showed less association with microtubules. And then the question becomes, okay, so if 20K likes microtubules and it likes mitochondria, does that microtubule binding, is it important to get to the mitochondria, or is it important once it's already there? And it ends up being the latter. Here's the 20K isoform with fully intact microtubule binding domain, and here's the isoform without the microtubule binding domain. And what you see is, at least to our eyes, the same kind of association with mitochondria, identified with a mito-BFP marker. Okay? So it really seems like, maybe this is not quite as tight an association, but I think that's stretching it when it comes to the limits of fluorescence imaging. Good enough for government work. It really doesn't interfere with mitochondria binding. Okay? So the 20K still labels the mitochondria with or without the microtubule binding domain. So does it affect the movement of mitochondria? And the answer actually to us is a pretty clear yes. All right? And so if you just look at this histogram, this is number of tracks versus velocity, because we clock individual mitochondrial movements. And what we see is that the black is in the control conditions. And then in the gray, we've used the cortisol. So we've taken away the detect microtubules. And what you see is the velocity just shifts to the left, right? The microtubules, mitochondria stop moving. But actually then, when we add 20K, we get enhanced movement, and very importantly, we see actually a subpopulation of mitochondria moving quite quickly. Just like those connecting vesicles that tended to hop on to microtubules and then zoom away. That's what we think we're seeing by this subpopulation. And when we use the DAL6 mutant of 20K, we lose this subpopulation. Okay? We shift back again to the left. So this is mitochondria are moving along microtubules, and 20K is not just facilitating their interaction, but it's therefore then facilitating the movement. And so the way we're thinking about it is 20K is basically the scaffold. It's this loading protein. It loads vesicles onto microtubules, and it can load organelles onto microtubules for movement. And then just a couple more slides where this is going is that here's the cell line. We're looking at just a mitochondrial distribution without and with oxidative stress in the presence of hydrogen peroxide. And what we see is that, in a cell, nice linear distribution and more peripheral distribution of the mitochondria, that as you add oxidative stress, they become more fragmented and centralized. Classic, right? Mitochondria fragmented and centralized when subjected to stress. Okay? In the presence of 43K, you don't see much of a difference, all right? They still centralize and fragment. Okay? In the presence of 20K, we see maintenance of the mitochondrial network, which implies, which David would teach me, it implies all kinds of energy efficiency and mitochondrial efficiency. Okay? And so we're rescuing mitochondrial network simply in the presence of 20K. And then when in the presence of del stakes, we lost that rescuability. Okay? And so how does this work into the heart? That actually, here's an example of ischemia, true ischemia, okay? Now in this case, we've done the same kind of mouse experiment that we did before, right? Injected AAV9, packaged 20K, right? We barely saw it on Western blot, all right? And this was actually, we went just under 4 weeks, and we had to use imaging to confirm that 20K is actually in the tissue. But even though we could barely see it in the tissue, we got really what to us is pretty profound protection to the myocardium. Okay? Here we had a really skilled surgeon tie off the LAD, left anterior descending artery, in the mouth, right? And then sacrifice the animals. And really, the sacrifice was just 3 days later, right? And what we saw is that if 4 weeks ago we introduced AAV, this is a viability stain where pink is viable tissue, white is scar. We lost a significant amount of the scar formation or prevented the scar from developing. We saved myocardium. And as you know, in ischemic heart disease, it's all about saving myocardium. And we can say, even though we subjected the heart to the same level of ischemic insult, we actually could preserve upwards of 30% of the myocardium with tinier bits, tiny amounts of 20K. And so in general, what really started out and still is for us, a trafficking-based story, right? We're seeing that actually the smaller isoforms have significant therapeutic implications. If they help connectin 43 get to the intercalated disc, and if you do it beforehand, before a heart is subjected to ischemia, such as during reperfusion, such as bypass surgery, such as any kind of major cardiac surgery, right? You may be able to limit the anticipated arrhythmias that happen subsequent to surgery, right? And in fact, go a little bit further, it's not just about acute electrical activity. What we're finding is that these smaller isoforms benefit the metabolic health of the heart. And by benefiting the metabolic health of the heart, they're actually protective. And protective in a way that we can actually, by modifying mitochondrial function, we can actually benefit them when they encounter ischemic episodes. And as you can imagine, it will be really interesting to know if we can benefit them long term in preserving myocardium from heart failure, which is something we've only just dreamed about. And so you can understand with that perspective, frankly, my excitement to see what's developing, because if these kind of therapies are developed, they're going to come out of here. I'm absolutely convinced, just to be very candid about it. So a great deal of appreciation to Jamie. You can see his work laid the foundation for much of what's happened since then. Shan Zhang is now at 23andMe, the genomics company, especially with the mitochondrial work. And there are folks in the group, I just want to highlight Wasim Bashir's role, but really Jamie actually helped recruit Wasim to the lab, and he's really evolved into really a talented experimentalist. Thank you very much.
Well, thank you, Rob. Astonishing.
Yeah.
Just great stuff. I'm going to come to you second, Jamie, but I'm going to go to Michael first. Michael, go.
So, thanks, Rob. Fantastic.
Yeah.
Really great stuff. One thing I'm curious about is either
from the genetics, from a human genetic standpoint, so looking looking at that region around the 213 AUG in terms of SNPs and mutations, not only could we find no record of a mutation, there actually was no record of a SNP. And so the lesson to us is that nature does not want that region modified. I mean, we just couldn't find anything that was modifying that region. And you know, there is this long-held belief, you know, is connection important or not to the heart, with a relative paucity of mutations in critical areas. And many of us, you know, believe that really what we see in the animal models occurs with human, that when you mess with key areas of connection, they're just non-viable, and so they don't make it out. And so we do believe that we're not going to find a human mutation in that. In terms of seeing it in terms of metabolic conditioning, to go, where we're seeing really is going is that we're quite astonished that although we saw connexin 43 full-length and small 20K isoform increased with preconditioning, increased with ischemia, when he subjected the animal to preconditioning, so when he subjected it to short bouts of ischemia before prolonged ischemia, 20K went up and 43 did not. And in fact, for those who don't know, Rob can probably explain this better than myself, but ischemic preconditioning in the heart and other organs, the bottom line is if you do a little bit of insult to an organ, you're actually protected from the subsequent downstream insult. And that's well known and that's well described, and there's a big debate of what mediates it. What we're seeing is that actually with the preconditioning, you get the 20K, and that actually is protective, and yes, actually, using the mouse model, we can actually replicate preconditioning protection just with 20K. And so that's still cooking, but it's a very exciting time, for the field for those reasons.
Go ahead, Jamie.
You're on the So my question is, 20K, stabilized Actin, cardiomyocyte, how much of that is fixing the connection properties, and how much of that is just
Yes, don't know. Yes, it's a great question. And were we incredibly lucky to find the one molecule that's important for trafficking in connexin 43, or is it sort of a general phenomenon that each has their own smaller isoform? And your guess is not only as good as mine, probably better than mine. We haven't gone there. Okay, you're familiar. I know all of us are familiar with connexin 43 being associated with something like sodium channel localization, right? And would love to go there, and but we literally haven't uncovered that yet. So I probably initially would have said each channel has its own subunit, but once we hit upon the mitochondrial work, it does seem like 20K, is more promiscuous than just its own protein. But how much it dominates and how much this is just a model for each protein to have its own subunit. I don't have a good answer.
Yeah, just the comment, Robin, just for the, like with many neural folks in the audience and on the ischemic preconditioning in the heart, the brain is also capable of ischemic preconditioning.
I've heard of it.
We have a brain, yeah. For us headless type
Yes.
stuff. And it's just interesting to speculate that if connexin 43 is central to ischemic preconditioning in the heart and other organs, connexin 43 is obviously very well expressed in many neural tissues, most especially the glial lineages.
Exactly.
Then it may well be that your discovery in the heart applies to the
May
brain as well.
very well. We'd love to believe so.
David.
I love the saying
Yeah.
that.
I'm in LA. It's an old Disney cartoon.
From an energetic sense,
Yep.
the saying is also that it inspires spoilers at the same time.
Exactly.
Right? Have you ever tried to inhibit mitochondrial function during the sort of expression phase, for example, to see if triggers the actual function of the mitochondria?
It's a great idea. No, we haven't tried that. I can tell you we have done seahorse assays, so that's not quite an answer, but we've gone in the other way. We've just started to look at the energetics. We haven't looked at ATP production, but we at least looked at oxygen consumption. And we got actually, just to be honest about it, we got a very surprising result. We expected these mitochondria to be super mitochondria and they can generate all kinds of ATP, right? But we got the opposite result. We actually saw a decreased oxygen consumption. And we'd love to measure directly measure ATP generation. And so, that's something we really do want to pursue. And so we just, I'm frankly need your guidance on that. But while that was initially surprising to us, it does fit the model of this preconditioning. Because clinically, it's well known that when a heart is subjected to ischemia, it actually becomes so-called stunned, right? And prolonged ischemia, it's hibernating, right? And the idea behind that is it actually doesn't work too well. When you reperfuse it, it comes back to life. And in fact, even though we're affecting the morphology, it may be inducing sort of this quasi-hibernating state. In that it's basically preventing, causing a low energy environment where it's decreasing ROS generation, preserving the health, preserving the survival of the cells until normal conditions can be established. So it's a little more complicated, but so you connect the big dots, it at least to us starts to make sense.
Randy.
Yeah. I'm sorry, therapeutic benefit and so there, you mean if it's applied ad hoc, right? Exactly. So exactly. So the first thought is it's going to have not much less of an effect than if you did it a priori. And that's why I very carefully said it's, expected ischemia as opposed to a sudden surprise, right? But let me throw two caveats out, there. The one is reperfusion arrhythmias, all right? The key reason there are ischemia arrhythmias are usually not the ischemia itself, it's the reperfusion, okay? And people have suggested a lot of that is to do with ROS generation, and I'll leave it at that. So, and so that's the one area that is actually an antiarrhythmic effect, whether or not it saves myocardium. But the second caveat is it might just darn well, for this whole reperfusion issue, may darn well actually be protective. So if the sun is ischemic, it's going to die. And in the heart, we say minutes of myocardium, right? But once you get revascularization, once you reperfuse, that's been the miracle of our percutaneous stents. When there's reperfusion, there's a growing body of evidence that actually a lot of the damage to the myocardium can actually occur with reperfusion. And so if you can get it in there at the time of reperfusion, you actually may provide a lot of benefit, in theory.
So I'm curious about the signaling pathways involved and how they link the connectin 43 molecule to the mitochondria. So you mentioned the mTOR signaling.
Yeah, yeah.
And which, you know,
Yeah.
links it to, I guess, IP3, AKT, you know, those various
So
other canonical
Jamie taught me, yes, yes.
pathways. So how does that relate to the mitochondrial story, do you think?
I think and now I'm starting to hand wave, right? But in, you know, sort of fed versus fasted state, because, you know, high up there is, you know, whether we're using insulin to induce a so-called fed state, right, versus fasted. Or, you know, another way of analogy of working at is sort of stress versus unstressed state. But by and large, you know, you are seeing the 20K up there during the stress, and I'm just trying to piece the data together with the clinical environment. I would only suggest it's there as a protective measure. Mm-hmm. And so that upregulation is not there to mediate damage. That upregulation is there to protect against damage. And definitely I would suggest that if it's not there, you can actually get greater amount of death and damage with a hand wave response.
And that's a more specific question.
Yeah.
So the 20kb isoform that had the microtubule binding domain.
Yeah, yeah.
Did you try that in the ischemia
No,
model?
no, haven't even tried that. Yeah, we did it for the we did it for the mitochondria work,
right.
And we really haven't moved the mitochondria work in vivo yet.
Right.
And so that's sort of on the drawing board.
Right.
Yeah.
That would start to indicate where along
It's a great suggestion. It's a really fantastic suggestion. Yeah. You know, really good.
Okay, Robin. Well,
Okay.
thanks so much. It's a wonderful talk, and it's so great
Thank
to have
you. Thank you for having me.